So the first question that really we need to ask is, what is the cytoskeleton? How can we define it? We can define it as really a simple class of proteins located in the cytoplasm that provide mechanical support and locomotion to the cell and the cell components. An important property of these cytoskeleton proteins is whether they are polar or not. This is roughly meaning that these proteins, through differencing in charges on their protein subunits, can provide directionality for the cell, indicated typically by saying one side is a plus side, one side is a negative side, which helps the cell know which way to shuttle cell components or move the proteins from one end to another for cell motility. Microtubules are polar, as well as microfilaments, also called as actin, thus providing these two proteins directionality for the cell to use. Intermediate filaments, on the other hand, do not have any distinguishable ends and are nonpolar. That doesn't mean that intermediate filaments aren't used by the cell. They are just not used in main methods for cell motility or moving cellular components around, as the cell doesn't know which one end is different from the other. Let's first look at intermediate filaments, as they are more simple in terms of the movement of their proteins because of their lack of polarity. If we go back to this figure here, we can first see the uniqueness compared to the others. Intermediate filaments have long twisting dimer proteins that bind together head to tail and tail to head to form an intermediate filament bundle. Looking more closely at intermediate filament structure, we can see, first of all, that is made up of subunits. These subunits bind to one another, forming a large intermediate filament. And because it's made up of subunits, we consider intermediate filaments biopolymers. Biopolymers are just biological proteins or chemicals that the cells make that are made up of smaller subunits to form a larger unit, very much like a polymer in your plastics. The basic function and structure of an intermediate filament is that it's made off of the monomer, which is a single polypeptide with the N terminus and the C terminus. Two monomers form a dimer, two dimers form a tetramer, and 8 tetramers form a protofilament. These protofilaments further coil with one another to form an intermediate filament. Now, because we can see that the heads and tails of the polypeptides that do have directionality and would be polar are binding to one another in an anti-parallel direction, heads to tails and tails to head, we form the non-polar structure, which is different from what we will see later on in other videos when we look more closely at the microtubules and actin filaments. There are 5 main types of intermediate filaments, with examples here shown. Type 1 and type 2 are acidic and basic, respectively, and examples of this is keratin. We can find keratin in epithelial cells, which help produce our hair and nails, and then other animals produce horns and scales. Type 3 is just known as type 3, and forms other proteins like desmin and vimentin, which is also important in sarcomeres in our muscle cells, in fibroblasts, in leukocytes, which are the immune cells, endothelial cells, which we can find lining our blood vessels, and other parts of the cytoplasm in different cells. Type 4 is typically called a neurofilament, and there's different examples of that, but we typically only find these in neurons and in their neuron axons to provide structure as well as strength for the axons during stretching and stress, mechanical forces to protect the cells from those types of movement. Type 5 is normally called nuclear lamins, and they're, as the name indicates, found in the nucleus. There's 3 main types there, A, B, and C. Moving forward, we'll be looking next at microtubules. I won't dwell too much time on microtubules. We'll go more in depth on them in a different video to talk about their polarity and how it's important to the cells here. But we'll look at the important functions that microtubules have in this section. Referencing back to this figure here, Looking at microtubules, we see that they're made of alpha and beta subunits. These are individually called alpha tubulin and beta tubulin. They form heterodimers, meaning they are made up of two different proteins that are formed together to make two dimers. They form long filament strands which come together to form the polymer, the biopolymer that is microtubules. The formation of this and importance of their polarity, again, I'll discuss in another video. So what are the functions of the microtubules? Well, looking at them, we can see, first of all, that they're important for the separation or organization of chromosomes during replication, such as mitosis and meiosis, as they are there in the nucleus binding to the genetic material, that's the DNA, the chromosomes, and help pull them apart to separate them into the new individual cells during mitosis and meiosis. Another important part of the microtubules is forming cell shape. A lot of this cell shape function that comes from the cytoskeleton is from the actin, but microtubules also have a vital role in producing the cell shape and size, as the cytoskeleton would provide the structure for it. One of the most important functions, on the other hand, of the microtubules that is distinct from the other cytoskeleton proteins is the shuttling of vacuoles and organelles. Because of the polarity and the strength and the rigidity of the microtubules and their larger size, it's perfect for other proteins to bind to and pull and move parts of the cell from one end to the other. Another aspect of microtubules is its assistance in cell motility. Actin is like the muscles. They will pull and contract inside the cell to move the cell, but in order to resist that contraction inside, the microtubules need to be there to provide that resistance, and this is called compression-contraction resistance, providing a platform kind of to push off for the cell to move forward and propel itself in a given direction. Lastly, let's look at actin. Again, there's a lot to talk about the importance of actin, but I'll just provide a brief introduction here and leave the more in-depth information to its own separate video. Looking at the structure of microfilaments, or actin, we see that it's made up of monomers called G-actin. Now, looking at it, you may ask, wait a second, how does this actin have polarity? It's only made up of monomers. Actin is very unique, and again, this will be part of a second video. But these G-actin monomers, when they bind to one another, they actually have ATP, part of the adenosine triphosphate, which we all know with metabolism, attached to them. When the G-actin monomers bind to one another, after a given time, the phosphate is hydrolyzed off, forming ADP on one end, and on the other end, the G-actin monomers still have their ATP bound to them. Therefore, this provides a positive and negative end indicated by the presence of ATP or ADP. So what are some actin functions in the cell? One of the most important functions that actin has is providing cellular movement. This movement is typically called actin treadmilling, and it's really just the polymerization and depolymerization of the actin microfilaments from one end to another in a dynamic way. One end could be faster or one end could be slower. I'll leave the detailed discussion of this in a separate video. Another thing that actin does is to provide structural support to the cell. One way that mechanobiologists see this function is in defining cell shape. Cell shape can be important for mechanobiologists because we've noticed that cancer cells typically tend to be more round versus the native tissue that is non-cancerous typically holds a defined shape that is non-cellular, such as cuboidal or fibroblastic. Mechanobiologists are investigating this link between cell shape and cancer to potentially provide an early indication that cancer is forming in that local tissue. Another way that actin functions in the cell is to provide protection for the cell from mechanical forces. Cells are not isolated from its environment, they experience mechanical forces all the time. Actin is great at protecting the cell from tensile loading and stresses and forces. An example of this is when you take a rope and pull it taut. That is an example of actin being loaded in an event that's tensile in direction to provide the cell structure. Now, if you compress the rope, it becomes weak. This is an example where microtubules per se would pick up the forces and protect the cell from those types of mechanical forces. One last note to make is that actin is in the nucleus, as well as intermediate filaments and microtubules. Now, actin has a different structure in the nucleus, but it's still there and has a vital important role of moving and organizing DNA. Researchers in mechanobiology have found that when actin has been mutated and knocked out the cell, we have decreased DNA repair mechanisms in place and see increase in DNA error, indicating that repair is not occurring. While there's a lot more required and needed to know about the function of actin in the nucleus, it's an interesting fact that actin is found there and is important for proper functioning. This kind of brings the point that actin and microtubules and intermediate filaments are in the nucleus, then there's probably a skeletal structure of the nucleus. And there is. Mechanobiologists are really interested in these cytoskeleton proteins found in the nucleus, which we have called the nucleoskeleton. This will be a whole separate video on its own, investigating the importance of these proteins in the nucleus, forming the nucleoskeleton. Thanks for watching. See you next time.